Hello, my name is Roy Morello, and I'm an associate professor in the Department of Physiology and Cell Biology at the University of Arkansas for Medical Sciences in Little Rock. My laboratory has had a long-standing interest in studying the effects of collagen mutation on the human body. We have done this using mouse models that faithfully reproduce the human disease phenotype. Our focus has been on type 1 collagen, the most abundant protein in our body. And type 1 collagen mutations cause osteogenesis imperfecta, also known as brittle bone disease or OI for short, the most commonly inherited bone fragility syndrome. OI can have a devastating impact on the skeleton, and its symptoms vary from mild to moderate to severe to perinatal lethal, depending on the underlying genetic mutation. OI is a systemic disease, and in addition to the skeleton, it affects other organs with variable severity. In the last 5 years, in collaboration with Dr. John Carroll, a pulmonologist at the local Arkansas Children's Hospital, we started to investigate the effect of type 1 collagen mutation on the lung, because newborns born with severe forms of OI can die of respiratory distress, but also adults with moderate to severe OI can have decreasing pulmonary function test and increased vulnerability to lung infections. While subskeletal features caused by OI, including chest wall deformities, can affect cardiorespiratory fitness, we and others have recently showed intrinsic primary lung defects in mouse models of OI. In our latest large study, we performed histological, histomorphometric, microcomputed tomography, and invasive studies on 3 distinct mouse models, mimicking mild to moderate to severe OI, with the overall goal of determining the extent of their pulmonary and respiratory mechanics defects and whether these defects correlate with the skeletal disease severity. Although with variable severity, OI lung histology consistently showed alveolar simplification with enlarged airspace and reduced alveolar surface. Numerous respiratory mechanics parameters, including respiratory system resistance and elastance, inspiratory capacity, total lung capacity, and others, were significantly and similarly impacted in mouse models mimicking a severe form of OI, but much less in models mimicking a milder form of the disease compared to control mice. Our data indicate that the impact of type 1 collagen alterations and OI on lung morphology and function positively correlate with the severity of the extracellular matrix deficiency. Therefore, it will be important to determine whether our observations in mice translate to OI patients and to dissect the respective contribution of intrinsic lung defects versus extrinsic skeletal defects to impaired lung function in OI. Thank you for taking the time to learn more about our study, published in the Journal of Physiology. The full paper was published in volume 601, issue number 2. Please feel free to contact me at rmorello@ums.edu if you have any additional questions.